Good afternoon. Am I audible?
Good afternoon, Doc. Yes po.
Okay, so can you start na? Sige. So there are 78 of you. Start na lang kaya it's 2:02 already. Okay. Sige, wait lang. Okay, so good afternoon, everyone. So yesterday, we started discussing about proteins, and if you can recall, so yesterday we deal about the biosynthesis of the non-essential or the nutritionally non-essential amino acids. So from that, we learned that non-essential amino acids may not be present in our diet because our body can or is capable of synthesizing this amino acid by using substrates from the citric acid cycle, from the glycolysis, and from other amino acids. So with that, for this as a continuation, this afternoon we will be dealing with the breakdown of proteins or the catabolism of proteins and what happens to the amino acid nitrogen. Kasi the protein, di ba, may carbon backbone, and it has an amino acid group. So in protein breakdown, so the bonds between the amino acid are broken down by peptides. Diba? So, and the individual amino acid are further broken down into the carbon backbone and the amino acid nitrogen. So to start, in the normal adults, nitrogen intake matches the nitrogen excreted by the body. So we have a term called the positive nitrogen balance. So there is a positive nitrogen balance when we have an excess of ingested nitrogen over what is excreted. So if there is an excess nitrogen, this can result in growth, and also this happens in pregnancy. On the other hand, we have the conditioned a negative nitrogen balance. So as opposed to the positive nitrogen balance, here the output exceeds the intake of proteins. So this usually follows conditions like surgery, advanced cancer, or in nutritional disorders, kwashiorkor and marasmus. So certain proteins, in the degradation of certain proteins, they uses certain enzymes, and they also use ubiquitin and ubiquitin ligases and deubiquitinating enzymes. Later on, we will explain what is the use of this ubiquitin in the degradation of some proteins. Ayun. So, the defects in the gene that encode these enzymes, yung ubiquitin, yung going back, yun. So, defect of the genes that produce this ubiquitin, these ligases, and these enzymes can result to conditions like Angelman syndrome, juvenile Parkinson's disease, von Hippel-Lindau syndrome, and congenital polycythemia. So that's why it is important for us to know at what level are the defects of the genes or of defect of the enzymes in order for us to understand these diseases or these defects. So in our body, the tissues in our body convert the ammonia, which is a highly toxic substance, to the amide nitrogen of the non-toxic amino acid glutamine. So, di ba yesterday, if you can recall, from an alpha ketoglutarate, we can add ammonia to produce glutamate. Also, from glutamate, we can add another ammonia to produce glutamine. So that is how our tissues convert this highly toxic ammonia as a part of the amino acid glutamine. And this glutamine now from the tissues goes to our liver. So it is carried by the blood to the liver, and there the amination occurs. And in the liver, the ammonia is again released, which is now converted to urea which is non-toxic, and the urea is the one excreted by the body. So if the liver function is compromised, so there are problems in our liver, like if there is cirrhosis or we have hepatitis, so there is now a problem in the conversion of the ammonia to urea in our liver. So what happens is that the blood ammonia levels will elevate, and this will generate clinical signs and symptoms of an elevated blood ammonia level. Okay, so first of all, let us understand so what happens to the protein in our body. So actually, protein turnover occurs in our body. Continuous degradation, so meaning breakdown, and continuous synthesis or turnover of proteins occur in all life forms. So it is said that in humans, this synthesis or this turnover is constitute 1 to 2% of the total body protein. So this happens most especially in our muscles. So that occurs each day. So the degradation or the breakdown of the protein occur in tissues that are undergoing structural rearrangement. So like, for example, in uterine tissue during pregnancy, so they they degrade higher rates of protein. So also in skeletal muscles in starvation or during starving state, as well as in tadpoles when they are having their metamorphosis. So approximately 75% of the amino acid from protein degradation, so when a protein is broken down, it is turned into the individual amino acids. So it is said that 75% of these amino acids are reutilized to make another protein or another substance, whereas the remaining excess free amino acids are not stored for future use, but instead rapidly degraded. So only the 75% are reused and the rest are degraded. So degradation of amino acid they are released as, so yung major portion niya, yung carbon skeleton, is converted to amphibolic intermediate. So they are being used in other pathways. And as I have said, the amino portion, so dinidivide ang amino acid from the carbon skeleton and its amino nitrogen. So the amino nitrogen is being converted to urea, which is excreted in the urine. Okay. So all proteins have what we call as the half-life. So when we say half-life, this is the relative susceptibility of a protein to degradation. So there is a certain time wherein a certain protein lowers its concentration to half of its initial value. So for example, liver proteins, they have half-lives from under 30 minutes to over 150 hours. Enzymes of the glycolysis pathway have a half-life of over 100 hours. And mga key regulatory enzymes naman, enzymes are also proteins. So these key regulatory enzymes have half-life of as low as 0.5 to 2 hours. So different proteins have different half-lives. And the degradation of these regulatory proteins with short half-lives and of abnormal or misfolded proteins occur in the cytosol. So the breakdown of proteins occurs in the cytosol of the cell, and this requires ATP and ubiquitin. So what is this ubiquitin? So as I have said earlier, so this is required in the protein degradation. So it is actually present in all eukaryotic cells. So it is a small polypeptide that targets many intracellular protein degradation. So it helps in the breakdown of proteins, ang ubiquitin. So that's the role. And this ubiquitin, so what they Yes. So what these ubiquitin do is they form bonds between its carboxyl terminal and the lysyl residue of the target protein. So they insert the ubiquitin molecules insert into the target proteins for degradation. And the residue present at the amino terminal affects whether a protein, so for a certain protein, there is a residue present at its amino terminal. So the type of amino acid that is present at this terminal determines whether a protein is to be ubiquitinated. So for amino terminals of methionine or serine, so this one they do not participate in ubiquitination. So unlike arginine, wherein there is an acceleration of ubiquitination. So when certain proteins are now ubiquitinated, so they are called as ubiquitin tag proteins. So yung mga ubiquitin, that molecule inserted themselves to a protein, so they are now subsequently degraded in the proteasome. So what is a proteasome? So a proteasome is actually a macromolecular cylindrical complex of proteins. So it looks like this, whose stacked rings form a central core that harbors the active sites of proteolytic enzymes. So as you can see, nakastack siya. So after a certain protein, ubiquitin attaches itself to that protein, and this now goes to the proteasome for degradation. So here, the protein must first enter, so the central core for degradation. So kailangan nasa center siya of this proteasome. So entry into this core is regulated by two outer rings. So this one that recognizes only polyubiquitinated proteins. So they do not allow the entry of proteins without ubiquitin. So kailangan it's an ubiquitin tag protein for it to enter this proteasome for degradation. Okay, so this is just another view of the proteasome where the protein degradation occurs. So, okay, so in our body, what the organ that plays a major role in the maintaining the circulating amino acid are muscles and the liver. So actually, it's in between these two organs, it is just ang nag-interchange lang from one amino acid to the other. So nag-transfer lang ang amino acid between these two organs. So it is said that the muscle generates over half of the total body pool of free amino acid. And here, so ang muscle is the one that generates the free amino acid, and the liver is the site of the urea cycle. So it is where the urea cycle enzymes are located, which is needed. So the enzymes of the urea cycle are needed for the disposal of the excess nitrogen. Okay, so if you can see this picture, so to elaborate on this picture, so we have here our muscles. So our muscles, as I have said, they release amino acid. So half of your amino acid pool in the body comes from the muscle. So they release this into circulation. So these amino acid are particularly alanine and glutamine. So glutamine and alanine from the muscle. The alanine is extracted by the liver. So it goes to the liver, whereas the glutamine is extracted by the gut and the kidney. So afterwards, the glutamine from the kidney becomes alanine going to the liver. So the glutamine is also a source of ammonia. So glutamine going to the kidney. So the glutamine, it undergoes deamination. So it now becomes a source of ammonia, which is excreted by the kidney. The kidney provides serine. So the kidney can form serine, which is later on goes to the peripheral tissues, the liver and the muscles. The muscles also can release branched chain amino acid in the form of valine, which is taken up by the brain. So by this, you can see that the amino acid just goes around between these major organs. Okay. So it is said that the alanine is the key gluconeogenic amino acid. So why it is the key gluconeo amino acid? Because here, diba, so alanine, diba, going back, alanine is produced by the muscle. So this goes out, going to the liver. When this goes to the liver, the alanine can become pyruvate, and from pyruvate becomes glucose, so now participating in gluconeogenesis. And here again, the glucose goes back to the muscles, breakdown again happens, and again alanine is formed. So it is said that the rate of hepatic gluconeogenesis from alanine is far higher from all other amino acids. So alanine is the key amino acid in gluconeogenesis. Okay. So here, this picture shows us what happens after feeding a protein-rich meal. So following a protein-rich meal, what happens? So from the gut or the splanchnic tissue, they release amino acids. So mostly the released amino acids are branched chain amino acid. So at the same time, the peripheral muscles also extract amino acids. Ayun. So which are also branched chain amino acid. So, okay, so that's how the protein turnover occurs in the body. So from one area, it is broken down. Then in another area, there is the formation of a new protein. So it's just parang a cycle in our body. So now we focus our attention to what happens with the ammonia or the nitrogen component found in the amino acids. So, but before that, so depending on their ecological niche and physiology, different animals excrete nitrogen as ammonia, uric acid, or urea. So all animals breaks down protein and they are able to make nitrogen. But how the nitrogen is converted into either ammonia, uric acid, or urea depends on the type of the animal. So like for example, in fishes, they are called monotelic. So because these fishes are in an aqueous environment, so they can use the water to continuously facilitate excretion of ammonia. So they are able to excrete ammonia as is. While in birds, birds are called as uricotelic. So uricotelic, they excrete the nitrogen-rich acid as semi-solid guano. So that is in birds. In humans naman, or in land animals, so including us, we are called ureotelic. So us, our nitrogen in ammonia is excreted as the non-toxic, highly water-soluble urea. So it depends on the organism on how we excrete the nitrogen component of the protein from the protein breakdown. Okay, so from that, we now go to the biosynthesis of urea. So how is urea formed in the body? So from the protein amino acid, it is broken down into the carbon skeleton and to its amino acid, to its nitrogen component. So that nitrogen component is in the form of ammonia, which is highly toxic. So we do not want that in our body. So what our body does is it converts this ammonia into urea to be excreted. So the biosynthesis or the conversion of the ammonia to urea occurs in 4 stages. So first there is transamination, followed by oxidative deamination. Then the ammonia is transported inside the mitochondria, and the urea cycle now happens. So there are 4 stages in making the urea. So starting with, let's start with the transamination. So if we can see here, transamination reaction happens. So there is an interconversion of alpha amino acid and alpha keto acid. So this is a freely reversible process. So all common amino acids except lysine, threonine, proline, and hydroxyproline participate in transamination. So from a one form, so it can interconvert, so it into the another form. So with uh, with ayun, so this is a freely reversible process. So one form can interconvert to another form of the amino acid. So as we have learned yesterday, the pyruvate from the glycolysis can be converted, or we can form alanine from pyruvate by using the enzyme alanine-pyruvate aminotransferase, or simply alanine aminotransferase. So that is one form of converting from one form of a substrate to another. So that is transamination. So another one is from alpha ketoglutarate. If we can remember, alpha ketoglutarate is from the citric acid cycle. So from that, we can form glutamate. So by using the enzyme glutamate aminotransferase, or glutamate alpha ketoglutarate aminotransferase. So that is also another form of transamination. From alpha ketoglutarate, we add ammonia, so now we form glutamate. So from here, it is said that alanine can also become a substrate for glutamate aminotransferase. So alanine can become a substrate. So it is, the alanine can be a substrate for glutamate aminotransferase. So that's why when we talk about transamination, we can concentrate on glutamate na lang, on what happens to glutamate, the glutamate amino acid. So now transamination occurs via a ping-pong mechanism. So hindi siya straight lang na one-sided lang na reaction. So it is characterized by the alternate addition of a substrate and release of a product. So ayun. So may papasok, then alanine, tapos pyruvate, so alternating lang from one substrate to another product, and another product to another, again, another substrate. So after removal of the alpha amino nitrogen of the amino acid, so what remains is the carbon skeleton na lang of the protein. And that carbon skeleton is also degraded by another pathway, which we'll talk about in our later discussions. So for this afternoon, we will just focus on the nitrogen component first of the amino acid. Okay. So in transamination, this requires pyridoxal phosphate. So pyridoxal phosphate is actually a derivative of vitamin B6. So this one is present at the catalytic site of all aminotransferases, and they play a role in catalysis, so or in the breakdown of the protein. So this, the role of this pyridoxal phosphate is actually they serve as the carrier of the amino group. So they carry the amino group during transamination. So by binding the amino group to the pyridoxal phosphate, what is created is a Schiff base. So the Schiff base is formed between the oxo group of enzyme bound PLP and the alpha amino acid of and the alpha amino group of an amino acid. So this Schiff base can rearrange, and so they are capable of rearranging themselves during transamination. So here, as you can see, so from transamination, which occurs in the tissues of the body, so actually it's like this. So there is a we are starving. So what happens is that or let's say we have intake of lots of proteins. So these proteins are now broken down by our body. So peripherally in our tissue, there is an increased amount of ammonia. So this ammonia are taken in by the tissue, and they are used in the transamination of the amino acid. So once this amino acid now are transaminated, or once the ammonia are now incorporated into the amino acid, this amino acid now is carried to the liver. So the nitrogen, so now from the glutamate, so the nitrogen is released as ammonia. So from the periphery na ammonia plus, so example, if we want to form L-glutamate, in the periphery. So what happens is that alpha ketoglutarate combines with ammonia. So alpha ketoglutarate combines with ammonia to form L-glutamate. So this L-glutamate now is taken to the liver. So it goes to the liver, and when it reaches the liver, it is now again the nitrogen group of this L-glutamate now is released again. So hinihiwalay na naman, and this is now the process of deamination. So they uses the enzyme glutamate dehydrogenase with use of NAD or NADP. Ayun. So from glutamate, as I have said, kasi we are nagde-deaminate na tayo, so we are removing now the ammonia. So what the product now again is back to alpha ketoglutarate. So ayun. So now ammonia is inside the liver of the is inside our liver. Okay. So here, the ammonia produced by enteric bacteria and absorbed into the portal venous blood, and the ammonia produced by the tissues are rapidly removed from the circulation. So this is what I have said a while ago, that the ammonia produced by the tissues, so produced peripherally, they are removed from the circulation and they going to the liver. So how are they removed from the circulation? By transamination. So transamination, and they go to the liver. And sa liver, they undergo deamination. So after nag-transaminate, they deaminate. And there, inside the liver, they are converted yung ammonia to urea. So should portal blood bypass the liver, systemic blood ammonia may attain toxic levels. So if there is a problem in our liver, so there is an impaired hepatic function, or there are collateral links between the portal and systemic veins, as in cirrhosis, so the ammonia in our systemic blood may reach toxic levels. And the symptoms of ammonia intoxication can be tremors, slurred speech, blurred vision, coma, and eventually death. So here, this is what happens in glutamine formation. So the glutamine synthase plays a major role in ammonia detoxification and acid-base homeostasis by forming glutamine from glutamate. So if we can recall, from L-glutamate, we can form glutamine, diba? From L-glutamate, as what we have discussed yesterday. And the glutamate can become glutamine by adding or by using ammonia and using the enzyme glutamine synthase. So this glutamine synthase now plays a role in balancing the acid-base homeostasis of our body. So there is a rare deficiency in the neonate wherein there is no glutamine synthase. So this results in, since they cannot convert the glutamate and they cannot use other ammonia to produce glutamine, so this results in severe brain damage, multi-organ failure, and death of the baby. So, glutaminase, so kanina it's glutamine synthase. So, glutamine synthase is for the formation of glutamine. On the other hand, from glutamine to glutamate naman, glutamine to glutamate, what the enzyme being used is glutaminase. So there are two types, the liver type and the renal type. So they differ with respect to their structure, kinetics, and regulation. So the hepatic glutaminase, so from glutamine, so in the liver, glutaminase level rise in response to high protein intake, while renal kidney type glutaminase increases in metabolic acidosis. So if there is a high protein intake, what happens? So sa periphery, there will be formation of glutamine kasi we want to get rid of the ammonia. So glutamine will be formed sa periphery. So which will be acted upon by hepatic glutaminases in the liver. So becoming glutamate. So that's why it is said here that in response to a high protein intake, the hepatic glutaminase level increases so that it can break down the glutamine from the periphery to glutamate. Whereas in the kidney, so the glutaminase increase in metabolic acidosis. So in the kidney, if there is metabolic acidosis, they want to break down the glutamine. So to release mga ammonia to make it basic. So that's why during metabolic acidosis, there's also an increase in glutaminase in the kidney to form ammonia by breaking down glutamine. Ayun. So here, excretion into urine of ammonia produced by renal tubular cells facilitates cation conservation and regulation of acid-base balance. So ayun, so this is now the role of the glutaminase. So they can regulate the acid-base balance in our kidneys. So the ammonia production from intracellular renal amino acid increases in metabolic acidosis and decreases in metabolic alkalosis. So ayun, ammonia increases in metabolic acidosis. So we want, during metabolic acidosis, we want to form more ammonia. So the glutamine is broken down by glutaminase. So during naman metabolic alkalosis, we want to decrease the amount of ammonia. So it's the other way. It's the opposite naman. So there is no breakdown of the glutamine. Okay. So from that, we now go to the urea cycle. So it is like this. So the urea cycle. The urea cycle occurs in our liver. So this one is the cytosol or the cytoplasm, and this one is the mitochondria. So from here, you can see that synthesis of 1 mole of urea, so this one mole of urea requires 3 moles of ATP. So where are these 3 moles of ATP? So 2 ATP here at the first reaction and another ATP here at reaction 3. And 1 mole each of ammonium ion, so this one, ammonium ion, and aspartate. And employs 5 enzymes. So 1, 2, 3, 4, 5. So there are 5 enzymes. So these are the requirements in synthesizing 1 mole of urea. So of the 6 participating amino acids, so here you can see there are several amino acids. The N-acetylglutamate, so this one actually functions solely as an enzyme activator. So this carbamyl phosphate synthase 1 cannot start or cannot function without N-acetylglutamate. So this amino acid here, its function is to activate this enzyme. Whereas the others, the remaining 5, are carriers of the atom. So ito siya, enzyme activator, while here, the remaining amino acid are carriers of the atom that ultimately becomes the urea. So while ammonium ion, CO2, ATP, and aspartate are consumed, so as you can see, ammonium ion, CO2, ATP, aspartate, so they are consumed in the urea cycle, ornithine consumed in reaction 2. So this one, this ornithine that is consumed in reaction 2 is actually regenerated in reaction 5. So here, from arginine, after the action of arginase, so here, what is produced now is the urea, and the other product is ornithine, which goes back inside the mitochondria. So the ornithine is not consumed, it is regenerated back in this cycle. So some reactions, as I have mentioned, some reactions occurs in the matrix of the mitochondrion. So this one, reaction 1, reaction 2. So this occurs inside. While the others, 3, 4, 5, they occur outside. Outside in the cytosol. So the urea cycle starts with here. So CO2, it is the condensation of the CO2. So carbon dioxide enters the mitochondria and the ammonia. So they have, there is a condensation of the CO2 ammonia with the help of the ATP and to form carbamyl phosphate. So this carbamyl phosphate is formed with the aid of this enzyme, carbamyl phosphate synthase 1, which is activated by this amino acid. So these steps are important because defect in this one, defect in carbamyl, and this one, the other enzymes, can result in metabolic disorders. So ayun, so yun ang dapat i-remember. So here, the first step is the formation of the carbamyl phosphate with the aid of carbamyl phosphate synthase activated by N-acetylglutamate. So after that, it is, by the way, so carbamyl phosphate synthase 1 is said to be the rate limiting of the urea cycle. So I have already mentioned this, it is only active in the presence of N-acetylglutamate. So this is the rate limiting step in the urea cycle, this number 1. So after synthesis of 1 carbamyl phosphate, so there is, to synthesize pala 1 mole of carbamyl phosphate, we require 2 moles of ATP. So one of this ATP serves as a phosphoryl donor for formation of the bonds, while the second, ATP, provides the energy in the formation of the carbonyl phosphate. So, ayun. So, phosphoryl donor ang isa is the one that provides energy. So, 2 ATPs are consumed. So, this transfer, the carbonyl phosphate is now transferred to the carbonyl group of the carbonyl phosphate is now transferred to ornithine. So ornithine is also inside the mitochondria. So the carbonyl phosphate, the carbonyl group is transferred to ornithine using the enzyme ornithine carbonyl transferase or ornithine transcarbamylase. So yun, name of the enzyme. And with that, when the carbonyl group is transferred to ornithine, what is formed is citrulline. So citrulline now goes out of the mitochondria into the cytosol. So it passes through membrane, mitochondrial inner membrane. So it passes through the membrane to go out into the cytosol. So now what happens to the citrulline once it is outside in the cytosol? So this citrulline now links with the amino group of aspartate. So there is, we use up aspartate and it links with citrulline with the help of arginosuccinate synthase. And this provides now the second nitrogen of the urea. So this reaction, again, also requires ATP. So this requires energy. So after that, the subsequent displacement now of the AMP by aspartate forms arginosuccinate. So from citrulline, there is the enzyme with the help of the arginosuccinate synthase, and aspartate is aspartate goes in with the formation of which eventually becomes the arginosuccinate. So this arginosuccinate undergoes cleavage using the enzyme argininosuccinate lyase. So cleavage, and when this cleaves off, the two products include arginine and fumarate. So fumarate. So the arginine continues here to reaction 5. And cleavage of this arginine by the enzyme arginase now forms urea. So urea, and the other product is ornithine, which again re-enters the mitochondria of the liver cell for another round of urea. So as you can see, cycle siya is the urea cycle from the ammonia. So from the ammonia, so it goes here, then again produce the ornithine. So continue lang. This is the cycle. Ayun. So the major changes in the diet can increase the concentration of individual urea cycle enzymes. So here, so these enzymes, this 1, 2, 3, 4, 5. So if there are changes in our diet, this affects the amounts of these enzymes. So like for example, in starvation state, the enzyme levels elevate to cope with the increased production of ammonia that accompanies enhanced starvation induced degradation of protein. Because during starvation state, diba? So what happens during this starvation state? The proteins, there is the breakdown or the degradation of the proteins in our muscles. And when that happens, so there is an increased production of ammonia because of that breakdown. So in response to that ammonia, what our liver do is increase the enzymes responsible for the urea cycle so that all this ammonia produced by the protein breakdown can be converted to urea because ammonia is toxic in the body. So in order for that to be released or to be excreted, it has to be converted to urea. So now we have metabolic disorders. So it is important for us to understand the steps of the urea cycle and the roles of each enzymes, because any problems in those steps or in those enzymes can cause metabolic disorders. So for the metabolic disorders, the general features include similar or identical clinical signs and symptoms can accompany various genetic mutations in a gene that encodes a given enzyme or in enzymes that catalyze successive reaction in a metabolic pathway. So if the problem is are among the genes that catalyze an that among the problem is in the genes that makes the enzymes in a successive reaction, usually the clinical presentation or the signs and symptoms of the individual are similar or identical. So a rational therapy is based on an understanding of the relevant biochemical enzyme catalyze reaction in both normal and impaired individual. So we should know what is the effect. So we should be able to compare the effect of a certain enzyme in a reaction that happens in a normal individual and compare it to the enzymes, the impaired enzymes in individuals with problems in with metabolic disorders, so that we can be able, will be able to know the therapy, so what to give for these patients. So number 3 general feature is the identification of intermediates of ancillary products that accumulate prior to a metabolic block provides the basis for metabolic screening tests that can implicate the reaction that is impaired. So by knowing what product is starting to accumulate in the body, we can know at what level of the cycle is there a block. So is there a problem in the enzyme? So that's why it's not going forward. So if there's a block, this the intermediates will start to accumulate, and that is the one that is being tested for metabolic screening. Then definitive diagnosis involves quantitative assay of the activity of the enzyme suspected to be defective. So for us to know the definitive diagnosis, we should be able to quantify quantitatively assay of the activity of the enzymes. The fifth general feature of metabolic disorder is that the DNA sequence of the gene that encodes a given mutant enzyme is compared to that of the wild-type gene to identify the specific mutations that cause the disease. So, ayun. And then the exponential increase in DNA sequencing of human genes has identified dozens of mutations of an affected gene that are benign or associated with symptoms of varying severity of a given metabolic disorder. So since we are now able to sequence DNA of humans, we can now better identify the genes that has mutations which affect the formation of a certain enzyme. Ayun. So this is just the table of the 5 enzymes found in the urea cycle. So we have the carbamyl phosphate. So from the reaction 1, then we have the ornithine carbamyl transferase when carbamyl phosphate and ornithine combines. So at the reaction 2, then we have the arginosuccinate synthase at reaction 3 when citrulline and aspartate combines to form arginosuccinate. Then we have the arginosuccinate lyase at reaction 4, wherein the arginosuccinate is broken down into arginine and fumarate, and the arginase wherein at the reaction of 5, wherein the arginine is broken down now into urea and back to ornithine, which goes back inside the mitochondria. So I would just like to highlight this because these enzymes are important in understanding the metabolic disorders of the urea cycle. So, ayun. So, urea cycle disorders are characterized by hyperammonemia. So, there is an increase of the ammonia because the cycle cannot proceed or cannot convert ammonia to urea. So there is hyperammonemia, encephalopathy, and respiratory alkalosis. Then 4 of the 5 metabolic diseases, so 4 deficiencies of the carbamyl phosphate. So from reaction 1, the ornithine carbamyl transferase. From reaction 2, the arginosuccinate synthase from reaction 3, and the arginosuccinate lyase result in the accumulation of precursors of the urea. So any defect or deficiency of these enzymes result in the accumulation of the precursors of urea, so which is the ammonia and glutamine. So ammonia intoxication is most severe when the metabolic block occurs at reactions 1 or 2. So sa may carbamyl phosphate synthase level and sa ornithine carbamyl transferase level. So when the block happens there, ammonia intoxication is severe. So again, clinical symptoms common to all urea cycle disorders includes vomiting, so avoidance of high protein foods, intermittent ataxia, irritability, lethargy, and severe mental retardation. So most dramatic clinical presentation of disorders of the urea cycle can occur in a full-term infant who initially appear normal and eventually becomes lethargic, and there is hypothermia and apnea due to high plasma ammonia levels. So the clinical features and treatment of all 5 disorders are similar. So this is what we have mentioned earlier, na they will present with hyperammonemia when there are problems in any of the enzymes needed in the urea cycle. So sometimes significant improvement and minimization of brain damage can accompany a low protein diet ingested as frequent small meals to avoid sudden increase in blood ammonia levels. So treatment of these metabolic disorders in the urea cycle usually begins with diet therapy. So the goal for this is to provide sufficient protein, arginine, and energy to promote growth and development, but simultaneously minimize the metabolic perturbation. So only low protein diet should be ingested so that there is no sudden increase in ammonia leading to hyperammonemia. So now we go to each of the defects for each of the enzymes. So here, to start, carbamyl phosphate synthase. So carbamyl phosphate synthase, as I have said earlier, this requires the N-acetylglutamate, the amino acid N-acetylglutamate as an activator. Without the N-acetylglutamate, carbamyl phosphate synthase cannot work. So this is the rate limiting step in the urea cycle. So the effects are responsible for the effects of this of this enzyme carbamyl is responsible for the relatively rare metabolic disease termed hyperammonemia type 1. So the effects of this enzyme result to hyperammonemia type 1. So next is we have the N-acetylglutamate synthase. So this N-acetylglutamate synthase is actually not one of the enzymes in the urea cycle. So this one is the enzyme used in the formation of N-acetylglutamate. So this catalyzes the formation of N-acetylglutamate from acetyl-CoA and glutamate. So why is this enzyme important? Kasi we want to form N-acetylglutamate, which is an activator for the carbamyl phosphate. So this enzyme also has an effect, the effects of this enzyme has an effect in the urea cycle if there is a deficiency of this enzyme, since no acetylglutamate will be formed. So deficiency of the said enzyme may respond to administration of N-acetylglutamate. So since the body cannot form N-acetylglutamate because of the absence of the enzyme N-acetylglutamate synthase, one way of treating patients who have this deficiency is to give them N-acetylglutamate na lang, the amino acid N-acetylglutamate to activate the carbamyl phosphate synthase 1 so that urea cycle can start. So next is the ornithine permease. So ornithine permease results from mutation of the ORNT1 gene that encodes the mitochondrial membrane ornithine permease. So if there is a deficiency of this enzyme, this can result to hyperornithinemia, hyperammonemia, and hemocitrulinuria syndrome, or the HHH syndrome. So this membrane, this enzyme is located in the mitochondrial membrane so that the ornithine from the cytosol can go back inside the mitochondria to be used again in another urea cycle. So failure to import cytosolic ornithine back into the mitochondrial matrix renders the urea cycle inoperable, with consequent hyperammonemia and hyperornithinemia due to accumulation of cytosolic ornithine. So ayun, so the ornithine must be able to go back inside the mitochondria with the help of these enzymes, or else the urea cycle will stop, and again there will be increase in ammonia and ornithine, so leading to hyperornithinemia. So in the absence of ornithine, what happens is the carbamyl phosphate carbamylates lysine instead. So instead of the ornithine, the carbamyl phosphate combines with lysine, forming homocitrulline. We don't want homocitrulline, but we want the citrulline to be formed to combine with aspartate eventually. So if this happens, there is the result of homocitrulinuria because of the absence of ornithine. So there's also a deficiency in the ornithine transcarbamylase. So here it's an X chromosome link deficiency termed hyperammonemia type 2. So this this hyperammonemia type 2 reflects a defect in this enzyme ornithine transcarbamylase. So Some mothers with this deficiency exhibit hyperammonia and an aversion to high protein foods. So patients with this deficiency have increased levels of glutamine in the blood, cerebrospinal fluid, and urine. So this is as a result of glutamine synthesis in response to elevated levels of tissue ammonia. So the glutamine level in the blood increases. Ayun. So another one is the arginosuccinate synthase. So arginosuccinate synthase is the enzyme that forms the arginate in the urea cycle. So without this enzyme, this results to citrullinemia. So there is the buildup of the citrulline in the plasma and cerebrospinal fluid. So here, about 1 to 2 grams of citrulline are excreted daily. Because the urea cycle cannot proceed because of the deficiency of this enzyme. So another condition, if there is a problem in arginosuccinate lyase, so this one now is the breakdown of the arginosuccinate. So what happens is there is a genosuccinic aciduria. So this is accompanied by elevated levels of arginosuccinate in the blood, cerebrospinal fluid, and urine. And this is associated with friable tufted hairs, also known as trichorexis nodosa. So there is an early and late onset types, and the diagnosis can be performed by measuring erythrocyte, argininosuccinate lyase activity on umbilical cord, blood, or amniotic fluid cells. Ayun. So here, the final enzyme needed in the urea cycle, the arginase. So defects of the arginase can result in hyperarginemia. So this is autosomal recessive, and symptoms usually appear at around the age 2 to 4 years. So from this one, the blood, there is, because the arginine cannot be broken down. So there is now an elevated level of arginine in the blood and cerebrospinal fluid levels. Ayun. So with that, if we are going to look back with those enzymes, so we can say that metabolic diseases caused by the absence or impairment of those enzymes can result to a very devastating effect. So usually, early dietary intervention can ameliorate the inevitable effects of this metabolic diseases. So that's why early detection is of primary importance. So there is procedure, the tandem mass spectrometry. This is a powerful and sensitive technique that can, in just a few minutes, detect over 40 analytes of significance in the detection of metabolic disorders. So this is a technique to know metabolic disorders in a baby, so that there is early detection of this metabolic disorders. Ayun. So, okay. So, going into the summary. So, for this afternoon, our topic was how is the protein broken down. So, it's the catabolism of the protein. And as I have mentioned, the protein has the amino group and the carbon backbone. So, these two is catabolized differently. So as of now, what we discussed about is what happens to the amino group or to the ammonia produced from this protein, wherein it enters the urea cycle to be excreted by the body. So ayun. So human subjects degrade 1 to 2% of their body protein daily at a rate that vary widely between proteins and with physiologic state. So Our body degrades the protein depende if we are in the fed state or in the starvation state. And here, key regulatory enzymes are used that have short half-lives. So proteins are degraded by both ATP-dependent and ATP-independent pathways. So we mentioned the ubiquitin earlier. So this ubiquitin binds to protein for degradation. So without for degradation inside the proteasome. So liver cell surface receptors also bind and internalize circulating as shallow glycoproteins destined for lysosomal degradation. Ayun. So proteins with polyubiquitin, so many ubiquitins attached to these proteins are degraded by proteases on the inner surface of a cylindrical macromolecule, which is the proteasome. And entry of these proteins inside the proteasome is gated by a donut-shaped protein pore that rejects entry to all but polyubiquitinated proteins. So we have mentioned this earlier na only the proteins with ubiquitin are allowed to enter the proteasome for degradation. So we also mentioned that different organisms excrete ammonia in different ways. So fish excrete the ammonia directly. Because of they are in an aqueous environment, whereas birds converts this ammonia to uric acid, and us, mga higher vertebrates, convert this to urea, a non-toxic substance. So transamination channels amino acid nitrogen into glutamate. So degradation of the ammonia in protein coming from the protein starts with transamination. So transamination, the amino acid is the nitrogen is introduced into to glutamate to form glutamine in the tissues. And after that, the glutamine synthase converts the ammonia to non-toxic glutamine. So the glutaminase releases ammonia for use in urea cycle. So after formation of glutamine, this glutamine now goes to the liver, and in the liver, the glutamine is degraded using the enzyme glutaminase, wherein an ammonia is again released for the use in urea synthesis. So in urea synthesis, there is a need of ammonia, CO2, and the amide nitrogen of aspartate. So these are the ones that provides the atom of urea. So hepatic urea synthesis takes place in part in the mitochondrial matrix and in part in the cytosol. So we have seen from the urea cycle that reaction 1 and 2 occurs within the mitochondria, and 3, 4, 5 occurs in the cytosol. So changes in enzyme levels and allosteric regulation of carbamyl phosphate synthase 1 by N-acetylglutamate regulate urea biosynthesis. So we also see that N-acetylglutamate is needed or required as an activator of carbamyl phosphate synthase 1, because without N-acetylglutamate, this cannot work, this enzyme cannot work, and therefore the urea cycle will not start. So metabolic diseases are associated with defects in each enzyme of the urea cycle. So each level of the urea cycle has an associated metabolic disorders. If there are problems in the enzymes for this for that reaction. So ayun. Also, metabolic diseases are also associated with membrane-associated ornithine permease. So if there is a problem in the ornithine permease, so the ornithine cannot go back inside the mitochondria. So again, this will result to metabolic diseases. And also, if problem in N-acetylglutamate synthase, problems of this one, so we cannot form the N-acetylglutamate needed by as an activator. So this will again lead to metabolic diseases. So all of these are needed in order for the urea cycle to proceed and so that there are no metabolic diseases in the individual. So metabolic disorders of urea biosynthesis illustrates 6 general principles of all metabolic disorders. So we have already mentioned that, and we have also mentioned the tandem mass spectrometry, so which is a technique of choice for screening neonates for inherited metabolic diseases so that we can treat them earlier. And like for this one, sometimes it's just the treatment is just dietary modification, so just lower the intake of proteins so that there will be no spike in the ammonia in the body. So lower protein intake. Ayun. So it's better if we can determine the metabolic diseases earlier so that they can be treated earlier. Okay, so I think that is the last of my slides. So, any question at this point? So actually, this is the highlight of our topic for this afternoon. So the urea cycle. And it is expected that we can identify these different enzymes involved in converting one substrate to the next in order for us to produce urea as an end product. So, any clarifications, questions? Doc, other inputs.
Dok, please po. Nakamita na ka mo, Dok.
Ay, ano yun?
Nagsasalita po ba si Dok? Ano man
I cannot understand, sorry.
is Doc Monsanto speaking to? I think he's moving.
Speaking to.
Yung
What is your question?
nakamute.
I thought you were saying something, Doc, kasi nakamit po kayo.
About?
Nag-think siya nga na istorya kanila. Kaya nag-movie mo mouth. Tapos nakamute.
No.
No.
Okay.
Yeah, I was
Now, this urea cycle, as you can see in this diagram, you'll be able to see that our urea cycle can produce arginine. And if you still remember, your arginine is essential. So it all depends on the period of your life where it is essential. As a fetus, it is essential.
Essential.
No? In fast-growing infant, that's essential. And then pregnant mother, that is essential. No? And in recovering from, especially surgery from traumatic accidents, no, that is essential because that would be required. That would be required for fast-growing children, for fetus, for pregnant women. And the amount of arginine produced by our urea is not sufficient. No, it is not sufficient at that particular period of time. But if you are an adult already and you have no problems with your health, arginine is now becomes non-essential in that matter. So that's why it's called semi-essential. So it all depends on the period of your life. No? Now, arginine is a very good, is a very versatile amino acid. I don't know if you know the story about Pfizer, in which they use arginine for trying to find out, because they were trying to, what you call this, they were trying to find a drug that would increase the amount of nitric oxide Nitric oxide is a vasodilator, and we can produce it. In fact, your nitric oxide is carried by your hemoglobin as well. Okay? Now, if now Pfizer was doing some research on it, until finally they were able to come up with an inhibitor in which it is used widely, and they come up with sildenafil. It is an inhibitor. It inhibits the destruction of cyclic GMP. Inhibition of cyclic GMP, in which it is needed in the synthesis of nitric oxide from arginine. Because cyclic GMP, I'm sorry, the enzyme that destroys the cyclic GMP is inhibited by that drug from Pfizer, which is sildenafil. So that's, but another thing with arginine, it can induce the eruption of herpes simplex. Herpes simplex. Usually herpes simplex, you'll find it will erupt around your nasolabial area. No? The nasolabial area. It is itchy, it is painful. That's herpes simplex. Now another amino acid that can prevent this, the blooming of herpes simplex because of arginine intake, is lysine, the amino acid lysine. So when you have herpes simplex, just take lysine capsule and they will disappear. The mechanism has not been known yet, but that is what they have observed. Now, we do not even know what is the reason really for eruption of herpes simplex with arginine. Then that's how it is, no? Now, some practitioners, especially in the integrative medicine or alternative medicine, they prescribe arginine as a adjuvant in the treatment of hypertension. So as an adjuvant, and the reason for that is arginine is acted upon by nitric oxide synthase so that it will produce nitric oxide. And nitric oxide is a very powerful, very powerful vasodilator. So the research of Pfizer looking for a treatment of hypertension led them to the compound sildenafil, which is very beneficial to patients having erectile dysfunction. So that's why they are making a lot of money out of it. Okay? Now, I remember when I took my exam, when I took my board exam, a question, urea cycle, I still remember this. Because as you can see, you're using only one ammonium molecule. No? One ammonium molecule. And when you look at your urea, there are two ammonium molecule there. No, two amino groups. Where did this amino group, this second amino group came from? You look at your diagram, your urea cycle. Where did it come from? See, from ammonium ion and carbon dioxide, you have carbamyl phosphate. Carbamyl phosphate reacts with your ornithine and you produce citrulline. You still have one amino group there. But when aspartate comes in, the aspartate now adds its amino group. And so you have it there. It becomes argininosuccinate. And this is being cleaved by its succinase, argininosuccinate lyase, I'm sorry, not the succinase, a lyase, and the carbon skeleton is fumarate, but the amino group is retained. And that's why you'll have arginine, there, 3 amino groups already. No? And then you have your arginase and releases it as urea. You have 2 amino groups there. No? It's additional. It is contributed by your aspartic acid. I missed that exam. I missed that number. I was not able to answer. So I cannot forget it. The additional amino group comes from aspartic acid. All right, as has been mentioned by Dr. Alita, the cycle, urea cycle, involves reactions inside the mitochondrion and reactions outside of the mitochondrion in the cytoplasm. Just remember that you have 2 reactions inside the mitochondrion and 3 reactions in the cytoplasm. Okay. I think that's the only input I can make. It was so detailed a lecture by Dr. Alita. And By the way, these lectures, what the presentation here, this PowerPoint, you can download this. You ask Sasha how to download it into your digital devices. It can be downloaded because I told Sasha to record all presentation. So you can download it if you want. Do you have questions? I have another question. Since during this pandemic, a lot of people went into gardening and they become plantito and plantita. And they found out that the usual fertilizer that is in the market contains urea. And they asked me if we can use our urine as a fertilizer. What do you think? Anybody there?
Yes, Doc.
No. Can you think of the reason? Can we use it as a fertilizer?
Yes, Doc. Pwede, Doc, gamitin, Doc.
Yes. Yes, but you have to dilute it. Why do you have to dilute it?
Masyadong concentrated po yung uric. Concentrated of ammonia.
No, it's not the ammonia, it's not the urea. It is the acid in your urine, which can kill your plants. So you have to dilute it with water, no? 1:1 ratio, so that you can use it as a fertilizer. The idea of diluting it is to dilute the acid in your urine. Now, I remember there was one time that there were in the cinemas, when you go to the CR, especially the CR for men, there are barrels there. And what are those barrels? They are for the urine, because there was one company who extracted urea from urine as a fertilizer. So they are retrieving the urine that is being shed in the CR of the male CR. But they did not pursue it because the owners of the theaters or those cinemas, and you know, when you store the urine there, it will smell. And so it was emitting an odor which goes inside the cinema already. So they cut off the putting of those barrels there. So the urea that you see here is the same urea that is used as a fertilizer. So if you are a plantito or plantita, you can save on your fertilizer by using your urine. Just be sure to dilute it, 1:1 ratio with water. Okay? So, Dr. Alita, you will be still lecturing Monday and Tuesday next week.
Yes, Doc.
Okay.
The catabolism of the carbon skeleton naman.
And specialized
Yes,
products.
and specialized products.
The porphyrins was not assigned to you, no?
Wala man, Doc. Doc, yung nakaschedule is Monday quiz.
You can have a quiz on Monday if you want.
Okay.
Or you can have it on Tuesday if you want.
Okay. Sige po, Doc.
Kasi you lecture only about 1 or 1.5 hours. We still have time for an exam. Or you give the exam before the lecture.
Okay, Doc.
Huh? Because if you give the lecture, then the exam, they will not listen to the lecture. Di ba? So you give the exam first and then the lecture.
Okay, Doc.
Okay. All right. That was a good lecture, Dr. Alita.
Thank you, Doc. So no more questions. Present question. Okay, so see you on Monday.
Thank you, Doc.
Thank you, Doc.
Thank you. Okay.
Thank you, Doc.
Thanks po,
Thank
Doc.
you.
Thank you, Doc.
Okay,
Thank you, Doc.
bye-bye.
Thank you, Doc.